Hi, I'm Madeline Ball, Director of Research for the Harvard Personal Genome Project, and I'm here to, thank you. A lot of you have probably gotten emails from me. I'm often interacting with a lot of our participants. I'm here to introduce you to our Explorers session of the conference. Science is exploration. As we explore, we break down barriers, the barriers between knowledge and ignorance, between theory and practice, and between researcher and participant. You might think the word explorers in this session refers to the scientists that will be speaking to you, and you'd be right, but you'd be half right. One of the most impressive and wonderful aspects of our project has been our growing involvement with these movements of citizen science and participatory research. Many of the speakers in this session are also involved with Get Labs, which many of you have seen downstairs, and they've invited PGP participants to join their research projects and further enrich their public research data that they're sharing with the world. We, participants as well as researchers, are all explorers. Our first speaker is Rob Knight, a leader in microbiome research. A professor at the University of Colorado, Rob Knight's group has played a leading role in developing open tools for microbiome analysis with the Human Microbiome Project. With the American Gut Project, they're taking their lessons into the world of citizen science with an exciting crowdfunding approach to bringing their science to all. Here's Rob.
Thanks, Madeline. And I'd like to thank Madeline and Jason for the very kind invitation to speak here. And what I'm going to tell you about, assuming this comes back up, is sorry, hard to type at this angle. How to make the HMP data work for you. So the issue is that when we're confronted with these large data sets, and I guess I revealed the joke a little early, is often when confronted with the data, we find ourselves feeling a little like this. And the issue is really that sequencing is getting so dramatically cheap. So much so that when Steve Quake sequenced his own genome towards the end of 2009, it was literally almost 100,000 times cheaper than it would have been in 2001 when the Human Genome Project was completed. And it's another couple of orders of magnitude cheaper today. So as a result, we're really experiencing sequencing in a very different way. Instead of a trickle of sequences escaping from behind the walls of a few major sequencing facilities, what we now have is this remarkable democratization of sequencing, where essentially anyone can produce sequence data. The machines are in the hands of pretty much any department, and soon in the hands of individual labs by this point. So then we run into this problem that we need to interpret all the sequence data and all these phylogenetic trees. So here I'm showing you just the first 9 out of 130,000 sequences from a recent project we did with Noah Ferrer's lab. Here's the first 0.1% of the tree file. Here's what happens if you just visualize the tree, right? And you can really tell you've got your work cut out for you. It doesn't even help you that much if I tell you the study design. So what we were doing was a biogeography study, but not on the grand scale of Darwin and Wallace and the other 19th century explorers who really delineated biological diversity on Earth, but rather a lot closer to home. So we got 3 people, sampled all 10 of their fingertips and all the keys on their computer keyboards where they type daily. And initially we were asking questions like, does the spacebar have more kinds of microbes simply because it's bigger than the other keys? The key's a desert where few microbes survive compared to the lush valleys of your fingerprints. And is there a dividing line, a Wallace line between the G key and the H key, where they're right next to each other on the keyboard, but you type on them with opposite hands? So anyway, another view of the data I just showed you makes the main thing going on immediately clear. So now each point represents an entire microbial community from a key or a fingertip. When I color those by subject, you see all the points from the first, the second, and the third person cluster together, but there's essentially no separation between keys and fingertips. So what you see immediately is the individual nature of the microbiome. Each of us transfers a unique skin community to from our fingertips to our keyboards as we type. And as part of the same study, we were able to show you could match the palm of someone's hand up to their computer mouse with up to 95% accuracy. So this came out in PNAS a few years ago, but more importantly, it was on CSI Miami, so you really know it's true. Anyway, so in the Human Microbiome project, the goal was to characterize what the healthy human microbiome looks like by taking 300 subjects, looking at 15 sites in each of the males, 18 sites in each of the females, and putting together 17,000 samples, 50 million 16S ribosomal RNA sequence reads, and a huge number of metagenomic reads and reference sequences. And so there are about 300 collaborators on this project, including Dirk Gevers at the Broad, who's here in the audience, and put the slide together. And so the HMP has produced this truly vast data set, both with 16S ribosomal RNA sequencing, which gives us a readout of who's present in each sample, and also in terms of shotgun metagenomics, where we can additionally do metabolic reconstruction, as well as doing a census of what organisms and what genes are there. And these are what was described in the two main papers that came out last year. And one of the main findings of the HMP is just how high diversity is at each body site. And one of the fascinating things is that no matter what body site you look at, for any taxon that's very common in one person, you can find someone else who has hardly any of it, even when you're looking at healthy people. So how we do the analysis and get from all those sequences to the kinds of visualizations I showed you for the fingertips and keys is with this open source pipeline called Chyme, which stands for Quantitative Insights into Microbial Ecology, where it's open source. It'll run on anything from your laptop to the Amazon EC2 cloud to the supercomputers at NERSC. And so what we do in the lab is we tag 16S genes from hundreds of different samples with DNA barcodes. We mix them together, and initially we sequenced on 454, although we've since moved to Illumina. And then because each sequence has a barcode, we can tell what sample it came from. So then we clip them off, we make alignments, we make phylogenies, and we cluster the samples where we hope those samples are going to cluster by phenotype. And so what it does is it makes these really noisy taxonomic signals understandable. So in a recent study we did looking at families, including the non-human members of those families, we start with really noisy data that look like this per sample in terms of taxonomic abundance, and turn them into these very clear cluster diagrams where, for example, you can tell that the dog's stool is much more similar to human stool than, for example, the dog tongue is to the human tongue in terms of its microbes. Incidentally, a lot of people wonder why the technology is called 454. So my collaborator Rick Bushman at Penn assures me that Fahrenheit 454 is the temperature your money burns at when you start doing this stuff, right? Sequencing's getting cheaper all the time, but it's still, it's still relatively expensive to do it on the scale we need to do it. However, the first time we did this at the end of 2007 cost us $12,000 and we got back 500,000 sequences. At the time, the sequencing facility down the hall from my lab was still charging $8 a read. So if we'd done it there instead of with the new technology, it would have cost $4 million instead of $12,000, and they wouldn't be finished with the sequencing yet. So I wouldn't be able to show you the rest of this stuff. And so this just reinforces the diversity numbers that I told you about before. So with Noah Ferrer, we looked at communities on the skin of undergraduate palms. What you see is we found about 750 species on 2 hands, about 500 on 3, about 300 on 4, and this tails off really rapidly, so there's essentially nothing that we find on more than 1/3 of the hands we looked at. And then the same is true in work we did with Jeff Gordon's lab at WashU and the gut, whether we use Sanger or 454, as we look at more people, we see less stuff shared among those people. And so Liz Costello in my lab was able to put together the first overall map of human body sites, where what we can see here is what we can see here is very clear clustering by body sites. So the mouth is very distinct, the gut is very distinct, the skin is a lot more spread out, and so forth. And so these are samples from 7 individuals who were willing to be sampled at 27 locations on the body, which, as you can imagine, is a lot of places to stick a Q-tip first thing in the morning. So this body site clustering is the clearest pattern. There's less of a signal by sex or by individual, with one exception. So one of our subjects mentioned on the day of sampling that had taken antibiotics a couple of weeks before for a sinus infection. And that person was totally different at every single body habitat from all the other people in our sample, although he was back in the distribution with everyone else 3 months later. So the study design was to look at everyone, wait a day, look at everyone again, then wait 3 months, and then do it again, so we could look over different temporal scales. And so this starts to give us the first picture of the overall variability in the microbiome with a lot of left-right symmetry. Here, a bigger bar means communities are more different. So samples from the same habitat, like both from the gut, are less different than those from different habitats. Within a habitat, samples from the same person are less different than different people. And then within a person and a habitat, samples a day apart are less different than 3 months apart. And then the mouth was the most stable, the skin was the most stable, and the gut was intermediate. And we also saw this in the HMP data set. But one thing that's a little frustrating about this is that in the Costello et al. Study, we only looked at 4 time points. And then in the HMP, we only looked at 3 time points. And to illustrate the issue with trying to look at dynamics that way, this is the viewer that we use to look at the results. So there's the gut, there's the mouth, and so on. If I rotate this around to give you an explicit axis for time, you can kind of see the 3-month interval. You can't really see the 1-day interval, but it's really hard to look at the dynamics, although you can kind of see some change. So what you'd really like to do is go back to some of the same people and sample them every single day for like 1.5 years. So with the Illumina technology, sequencing was finally cheap enough that we could do that, and it looks like this. And the concordance between the new data on Illumina and the original data on 454 is just beautiful. And Antonio Gonzalez, a really talented CS grad student in the lab at the time, figured out how to animate this. So what I'm showing you here is 6 months in the microbiome of 2 people, looking at the skin, the mouth, and the gut. So the orange point over here is me. And what you can see is you can just immediately see for yourself how much variable the skin is, how static the mouth is, and how the gut's intermediate. And you can also imagine that tracking people over time who have interesting clinical conditions, like, for example, IBD, with cycles of remission and relapse, or if you're doing fecal bacteria therapy and trying to cure C. diff, for example, by transplanting the microbes, it could be really effective to trace those things by looking at these detailed time series. So temporal resolution is really useful. So is spatial resolution. So this is Chris Lauber, one of my current postdocs, who was interested in microbes on the human face. So he decided to begin by looking at his own face, which he divided into 50 different patches. And using the techniques I showed you, you can kind of see some patterns. So those 2 patches are really different. Those are the lips, and I told you the oral communities were really different from everything else. And there's kind of some clustering because the patches are numbered sequentially, but it's difficult to figure out what's going on until you paint the data directly on the face itself. So for example, if I show you PC1 of the microbial community, you immediately see the left-right symmetry. If I show you PC2, you see this one patch that's different from everything else. It turns out that our subject wears glasses and tends to adjust them from time to time, and the fingertip communities are pretty different from the communities on the rest of the face. And then the third axis, basically what we're seeing is the separation between the lips and everything else. But we can also match this up to taxa that have names that you can recognize and so forth and paint them on the face too. So for example, if I show you Propionibacterium, a classic skin bacterium, there's a ton of it everywhere in red except the lips where it's washed out by the oral community and the ears which stick out a bit and they're a bit colder than the rest of the face. So that's restrictive for the growth of this taxon. And then this Prevotella, which is a classic oral bacterium that you see a ton of on the lips. And then you see just a little bit in one of his ears and also on one of his cheeks. And you can come up with your own scenario for how those oral bacteria got there, right? So of course, what this means is that with more detailed time series, we start to get at the behavior of our subjects. This raises all sorts of ethical issues that I really shouldn't tell you how we do recruiting for this stuff. So you might be wondering where our microbes come from in the first place. And if you have dogs or kids, you probably have dark suspicions about that. And it is in fact true that dogs share more microbes with their owners than other people. You can even match the dog to the family by looking at the microbial communities. Then with Maria Gloria Dominguez at the University of Puerto Rico, we were able to sample the microbiota of mothers an hour before they gave birth, and then of the kids 20 minutes after birth. And so in babies, all of the body habitats look the same instead of being differentiated as they are in adults. And basically, if you come out the regular way, you look like your mother's vaginal sample. If you're delivered by C-section, you look like skin. And here's just another view of the same data, showing the vaginally delivered babies clustering with the vaginal samples, and the C-section babies clustering with the skin samples. And then the mother's oral community is really different. And then we can also trace development in infants. So here, this is work with Ruth Ley, where we're starting with the meconium of one child, looking at weekly fecal samples over the first 2.5 years of life, during which they come to resemble the mother, also with a steady increase in diversity. But what's really powerful at the moment is that we can start using the Human Microbiome Project data as a data frame to understand these other data sets. So when we do that, what you can see is that the baby gut starts off looking like a vaginal community, which is consistent with the delivery mode of this individual. So each step in here is a week, and you can see them bouncing back and forth between the skin and the vaginal community in green and pink respectively. And what happens is that they're starting to get more and more like the adult stool, which are these samples over here in brown. And one thing that's interesting coming up in just a moment is that the kid gets antibiotics for an ear infection, and then you see this, so it's coming up right now, you see the sudden and very radical change in the microbiota, but then you see a fairly rapid recovery over a few weeks. And by the time we get up to 27 months, you can really see them resembling the adult stool. So by using the HMP data as a data frame to interpret these other data sets, we can really get a lot out of it. We also see this very reproducibly across cultures. So when we look at people from Malawi, from Venezuela, and from the US, the trajectory of how rapidly the infants develop over the first few years is very consistent, although the end state of the populations is very different. And in this context, it's really important to remember that in the HMP, we only capture healthy adult Western populations. We don't see all the rest of this variability that we still need to characterize. So the goal of projects like American Gut is to really reach out to the public to capture different ages, different populations, people of different health status, and so forth. And so Daniel McDonald, one of the very talented IQ Bio students at Boulder, who's putting in a huge amount of effort at American Gut, is downstairs manning the kits there, and we'd like as many of you to sign up as possible and contribute samples. We've got over 4,000 people already, including 400 samples from the PGP, but we definitely need more. I see I'm out of time, so I'm just going to end this by showing you where we are with the PGP data and the American Gut data at the moment. So the blue samples are from a class at Hopkins who signed up to give oral samples. The red samples are from the PGP, and you can see that we have a whole ton of information about the kids from Hopkins who filled out the American Gut consent forms and questionnaires. But from the PGP subjects, we don't have any of this information. You can see that it's always just coming up as NA in those big clusters there. And so what we really need you to do is we need you to claim your data and to start being able to understand what the reason for the variability is in these different samples, so that we can really get somewhere in understanding what alters the microbiome and what has a large effect and what has a small effect. So with that, I'm just going to finish there. Thank the large number of current and former members of my lab who've contributed to this, our collaborators on various projects, especially Noah Farah, Jeff Gordon, and Ruth Ley, whose work I showed you, the 300 or so people in the HMP, and then the people involved with the Earth Microbiome Project and American Gut, and our various sources of support. So thanks for your attention, and I have a few minutes for questions, which I'd be delighted to take some of.
So when you're looking at, the babies after being born, have you looked at like breastfeeding babies, like how the mother's nipple microbiome's different and how that might interact with a breastfeeding baby?
Yes, and so one thing I skipped over at the end there is this Gates Foundation-funded project where what we're doing is we're taking microbes out of healthy and malnourished twins, putting them into sets of mice. So this is work with Jeff Gordon's group at WashU and a number of other labs around the world. So putting those microbial communities into mice and then seeing how the mice are able to maintain their weight or whether they lose weight depending on whether they get the sample from the healthy twin or the twin with kwashiorkor. And we're also tracking those individuals enrolled at birth every month for the first 2 years of life. So fascinatingly, we had assumed that you'd see a large discontinuity with things like when you switch from breast milk to solid food or from breast milk to formula. We don't really see that. What we see is that an overall maturation pattern is the strongest signal. So individual infants who stop breast milk earlier or later tend to look the same as individuals of the same age rather than individuals of the same diet. And you have to do much more sophisticated analyses to pick up the subtle effects of breast milk. Now, that doesn't mean that those effects aren't there. There are statistically significant differences, but a lot of what we're trying to do at the moment is to figure out what has a large effect on the microbiome and what has a small effect, and especially which of those effects are correlated not just with changes in the microbiome, but also with human health. So breast milk, for example, gives you lower rates of a range of allergies, autoimmune disease, and so forth, although again, you're looking at effects that are statistically significant, but they can be small compared to other environmental factors.
I'm curious about how the microbiome changes as we travel, and if you've done studies on that, and if you can imagine implications for preventive strike on travelers' health.
Right. Again, that's a great question. So we don't have a large-scale study. 4 of us, including Antonio, who put together the awesome time series that you saw, went to Bangladesh to do a cloud computing workshop in Dhaka last year, and we tracked ourselves daily during that process. And our microbiome certainly changed substantially during that period, especially after we'd been eating the food for a few days and got sick. So that's something that would be fascinating to do on a larger scale, especially to figure out the extent to which we can recover where someone has traveled based on the microbes that they're carrying around inside them and how long those effects last. So you've got about 100 trillion microbial cells in your gut, right? So they outnumber your human cells by a factor of about 10 to 1. On average, they have genomes that are about 2.5 million bases long. So if you do the math, there's a whole lot of data sitting in there waiting to be decoded that probably has a whole lot of information about your medical history, your environmental history, and so forth. And it presents a tremendous opportunity. So.
Hi, Rob. Yeah, it's a great talk. Sorry to cut you off.
That's okay.
Yeah.
Thanks so much.